cost. And so this is exactly what I expected to find. When I look at the same relationship with the lifetime reproductive success of the mom, the number of pups that she produced over her life, I don't find any correlation, which is a little bit surprising. But I will note that this reproductive success measure does not yet incorporate her pups' overwinter survival. And so perhaps if I were to look at the number of grand offspring she has, or the overwinter survival, or the number of pups that recruit into the population as adults the following year, then we might see a stronger correlation between her reproductive success and her endocrine plasticity. Nevertheless, this suggests that red squirrels differ pretty drastically in their ability to track changes in local density, and mums that lack this ability to adjust glucocorticoids appropriately pay a cost as far as having a shorter lifespan. So far, I've shared with you a story of a solitary animal where the social environment essentially represents a stressor, but that's not always the case. And in fact, in a lot of unpredictable environments where conditions fluctuate really drastically, animals tend to be more social. And so there's a hypothesis, or there's a pattern, that cooperative breeding species and that group living or family living species are found more commonly in these unpredictable environments that fluctuate a lot. This is true in birds and mammals and even in human societies. And so I next want to share with you a story about how social living can actually represent a potential coping strategy for unpredictable environments. And so this is work that I conducted in the superb starling, which is an East African passerine bird that is highly social. These birds live in really large social groups that range from about 20 individuals all the way up to 60, so they can get quite big. They're also a plural cooperative breeder, which means that within one social group, multiple breeding pairs can initiate a reproductive attempt during the rainy season, which is their breeding season. At each nest, there will also be between 1 and 15 helpers that contribute to caring for young. And these helpers can be both related or unrelated to the young. They can be both male and female. What is really interesting about the superb starlings is that they live in groups that are significantly larger than what would be needed for helping at the nest alone. And what I mean by that is about 50% of birds in a social group will not partake in breeding whatsoever in any given season. So they won't breed, they won't initiate their own independent reproductive attempt, and they also won't help at an available nest. And so this led me to believe that group living is actually potentially important for reasons beyond cooperative breeding. This is not all that common in a lot of birds, where the size of the social group tends to be the breeders and the helpers. And so we have all these extra individuals in superb starlings that belong to social groups but don't reproduce or help whatsoever. And so I hypothesized that these individuals might serve to buffer against harsh conditions by improving survivorship of adults. So I'm using here a long-term data set that began in 2001, where individuals from 7 social groups have been uniquely marked with metal leg bands, but also colored leg bands. And we've also monitored over 800 nesting attempts in each of these social groups since 2001. And this is all work that's conducted at the Impala Research Center in central Kenya. Superb starlings experience extremely variable environmental conditions in 2 ways. The first is temporal variation in rainfall from year to year. And so there tend to be 2 rainy seasons that happen every year in Kenya. And during these rainy seasons, the birds tend to breed. But the amount of rainfall that falls in the rainiest season of the year tends to fluctuate really drastically from one year to the next. And so some years have really high rainfall, like 4-fold increases relative to the average. And then some years have very, very dry rainfall. So even the wettest part of the year is nearly all dry. The other way that the habitat fluctuates is spatial variation in grass cover. And so the bottom picture here is an aerial photograph of the research center where I worked, but you can sort of see that vegetation is quite spotty. And so grass cover varies from one territory to the next. So social groups experience variation in grass cover based on which territory they're defending. And so what rainfall and grass cover together do is shape the availability of insects, which is the #1 source of food that adults prefer to eat, and it's the exclusive food that they use to raise their young. And so these insect prey are really important. So because they live in such big groups, I hypothesized that living in a big group would improve the survivorship of adults living within that group, but I expected that group living would buffer against harsh environments. And so what I mean by that is there should be a positive correlation between the size of a social group and survivorship of adults, but that relationship should have a steeper slope in harsh years or on territories with low grass cover. And the benefit of being in a bigger group would be less in a benign year with high rainfall or on a territory with high grass cover. And so to test this, I used Cox proportional hazard regression models. And here you can see a survivorship plot. So the survival probability is on the y-axis, and 1 means 100% of birds were alive. 0 means 100% of birds were no longer alive. The x-axis shows you the age of starlings in years. So you can see they're quite long-lived. Many individuals live past 12 years of age. First, I'm going to share with you data for female starlings. And I've got 3 survivorship curves, which are the thick lines displayed here. The pale gray line corresponds to individual females that lived in the smallest social groups. The dark gray line shows individuals in the average social group sizes. And then the black line represents individuals in the largest social groups. And so what you can perhaps see from this figure is that individuals in the largest groups, that black survivorship curve, have improved survival relative to individuals in the smallest group, that pale gray line. And so there's a clear benefit to being in a bigger group where adult survivorship is higher. For the females, I'll say that I didn't find any correlation between the benefit of being in a bigger group and any environmental conditions. So there was no interaction with rainfall or grass cover. And so this benefit of being in a big group seems to apply regardless of what environments females are facing. When I look at the same pattern for male starlings, I actually find that there is a significant interaction between group size and rainfall. And so here I've separated the figure into 3 panels that differ based on the rainfall conditions that males were facing. But the legend where the smallest group is the pale gray line, the average group is the dark gray line, and the largest group is the black line remains the same. And so if we focus on the panel on the left-hand side under harsh years, panel A, you can see that there's really no difference between survivorship of male starlings and group size. And so males across all groups have survivorship that's not significantly different from one another under harsh years. In the middle panel, under average years of rainfall, I similarly don't find that there's any benefit of being in a bigger group. Survivorship is simply not different for individuals based on their group size. In contrast, on the right-hand side, in high rainfall or benign years, I do find a pattern where there is a benefit of being in a bigger group, where males in the largest groups have higher survivorship relative to males in the smallest groups. And again, this is only a benefit that is expressed under high rainfall years. And so this is actually the complete opposite of what I predicted. I expected survivorship would be most beneficial. Survivorship would be higher in larger groups under harsh conditions, but I found that under favorable conditions, superb starling males benefit most from being in a bigger group. And I only have one main hypothesis for why this might happen, and it's that under harsh years, there's just more competition for breeding positions within a social group. And so males in a large group might face more competition, and that competition might be more fierce under low rainfall years. And so that benefit of being in a bigger group might sort of be counteracted by increased competition within your group when conditions are harsh. Under years of high rainfall, males might benefit from being in a big group because that competition is relaxed and more males are able to breed when there's more rainfall. So that might be why I didn't find support for the specific hypothesis I was testing. Overall, though, there's a clear benefit of being in a bigger group where starlings from larger groups, both males and females, do live longer lives. But it's unclear why this pattern happened. So why is there this link between survivorship and group size in this species? And there are 3 main hypotheses to explain that. The first is that individuals in the largest groups have better access to forage in some way. The second is that individuals in larger groups are somehow in better or superior health or condition. So some examples are that some species in larger groups have better immune defenses or have reduced physiological stress, for example, from sublethal effects of predators. So being in a bigger group can be beneficial for an individual's condition. The third hypothesis is that predation risk drives differences in survivorship, and so individuals in the largest groups face reduced predation risk. And so I'd like to go through some data that I have to address these three hypotheses. First, the idea that bigger groups have access to more food. So in superb starlings, that doesn't seem to be the case. The two environmental factors that shape food availability, rainfall, and grass cover were not correlated to the survivorship of adults. And so starlings that experience more food availability in their environment do not have longer lifespans necessarily. And so that suggests to me that there's not really a clear connection between food access and survivorship. The other thing is that starlings rarely hang out in their large social groups all at once. They tend to hang out in subgroups, and they also forage off territory a lot, so they don't really seem to defend food resources as a social unit. And so that doesn't really support the idea that being in a bigger group gives you better access to food. One alternative is that being in a bigger group improves the condition of individuals within that group. And so to do this, across 4 years, I caught individual starlings from multiple social groups that differed in size, and I assayed different physiological measures. So I looked at a couple measures of glucocorticoids, both baseline and stress-induced. I looked at a few measures of oxidative balance, both reactive oxygen metabolites and antioxidants. And then I looked at bacteria killing ability of plasma to standardized E. coli strain, and I looked at body condition of individuals. And I'll tell you really quickly, most of these traits didn't co-vary with group size across individuals, but I did find 2 traits were correlated with group size. So I'll tell you more about those. So the first trait that was correlated with group size was baseline glucocorticoids, and this was in a nonlinear way. And the relationship was different for breeders and helpers. So here I've got breeders as the open symbols, and I've got helpers as the closed symbols. And so what you can see is that there's this quadratic relationship between glucocorticoids and group size, where individuals have lower baseline glucocorticoids at intermediate social groups. Similarly, antioxidant capacity was correlated to group size in a nonlinear way, and individuals in the smallest and intermediate groups tended to have higher antioxidant capacity, which is beneficial. And so what both of these things suggest to me is, as far as an individual's condition goes, intermediate group sizes are best. And so to optimize your condition as an individual superb starling, you should opt to live in an intermediate size group. So this doesn't support that an individual's condition is greatest under the largest social group sizes. The third possibility is that larger groups better defend themselves against predators. And unfortunately, because the superb starlings don't all hang out together, it's really hard to study these sort of vigilance benefits of group living. We do know that adult starlings do get eaten by a number of predators, including snakes and birds of prey, which are abundant in the landscape. But we just don't have really clear data on how different social size groups might respond to predators in the adult context. What we do have a lot of information about is how predators at the nest influence reproductive success of the group. And so we do know that over 90% of nests of superb starlings get eaten by predators, which is quite high. And we also know from a predator model experiment where either a model bird of prey or a model snake was presented at the nest that all group members contribute to mobbing the predator cooperatively. And what I mean is, yes, the parents will mob, and yes, the helpers will mob, but all the other group members will also contribute to mobbing the predator if they hear an alarm call from their group members. And so this suggests that being in a bigger group gives you access to a bigger pool of individuals that could mob predators cooperatively. Superb starlings even engage in this guarding behavior where they spend a lot of time perching near the nest in order to alarm call when a predator does approach. And so this guarding behavior should be really important to improve survivorship, at least at the nest, if predators are one of the main sources of nest failure. And so this is something that we see. Here I have the likelihood of fledging at least one offspring from a nest on the y-axis, where 1 is one offspring was successfully fledged, 0 is none were fledged. And then here on the x-axis, I have the total cumulative amount of guarding that individuals performed at a nest. And what you can see is that nests that are better guarded are more likely to be successful. So altogether, these results suggest that larger groups do not have access to more food. To a certain extent, larger groups do improve the condition of individuals, but in the largest social group sizes, individual condition starts to decline again. And then, at least at the nest level, it does seem that being in a bigger group could give you access to more individuals that could cooperatively mob predators. And this is likely to be the same at the nest and for adults. What I did notice is there's a ton of variation in guarding behavior, which is the #1 way that individuals can defend their nest against predators. And you're not always going to be successful, but it's the strongest predictor of nest outcome or nest success. And so I was really interested in the factors that shape variation in guarding behavior among individuals. So why does one starling choose to guard a lot, and why does another guard less? And so I have a fantastic undergrad student at Kennesaw State who was interested in this question, and she thought that guarding was a factor of how much time you had available after you took care of other activities. And so she was interested in the idea that individuals experiencing high rainfall would be released from having to search for more food, and this would allow them to guard more at the nest. And so in this way, under years of higher rainfall, individuals should be able to increase their guarding. And so she used a reaction norm approach similar to the approach that I used in the squirrels. And here I'm showing you guarding behavior on the y-axis and breeding rainfall on the x. And we've separated the figure into breeders and helpers. And so what you can see if you look at the blue lines, which are the population average, is that at least, especially for breeders, there's a positive correlation between guarding and rainfall, where there is more guarding under higher rainfall. But if we focus in on the individual black lines, which each represent one starling, there's a lot going on, and there's no clear pattern where an individual tends to increase their guarding under higher rainfall versus some individuals decrease it. So this is not a clear pattern like with the squirrel system, for example. And what Jasmine found through random slope models is that the best model to explain the data that we have suggests individuals differ in their guarding behavior on average, but there's no clear plasticity where some individuals change guarding behavior relative to rainfall. And so this suggests that the pattern that we see where there's more guarding under high rainfall might actually just be driven by who helps under high rainfall. And so some birds that are likely to guard more might be the ones that choose to guard under high rainfall, and the individuals that guard less might be the ones that choose to guard under low rainfall. And so this suggests that individual starlings might differ in their guarding behavior, but they don't seem to show plasticity in response to changing rainfall conditions. And I suspect that one other environmental factor that might be really important for these starlings in shaping guarding decisions might be what other individuals in their group are doing. And so the social environment might be a really important part of this decision, and this is a direction that we're heading in the future. So all of these problems that I've worked on in the past have really got me interested in why individuals differ in their response to the environment. And from an applied perspective, I'm really interested in how you make an individual more flexible or more resilient. So can you manipulate something about an environment's upbringing or an animal's upbringing that makes them more flexible? And so this is the type of work that we're tackling in my lab now. And we're studying European starlings, which are common everywhere, but especially near our campus. And so while European starlings certainly don't experience the same types of rainfall variation that superb starlings do in East Africa, they do experience a lot of different environmental pressures based on where they are. And so we're studying 3 populations along an urban to rural gradient where birds experience differences in human density, but also in the availability of food waste related to humans. And so today I want to share with you some preliminary data from 2 sites, a rural farm and an urban farm. The rural farm is out in the county, and it's a cattle farm with a lot of corn fields where starlings can find lots of food and insects. The urban farm is a really cool site because it's a similar farm property where insect availability is really high on the site, but right across the street, starlings can choose to go hang out in a McDonald's dumpster if they choose to. And so in this way, the urban farm represents an area where starlings can choose to live a more urban lifestyle or a more rural lifestyle. And so urban birds are generally thought to be more flexible in a lot of ways. They respond more flexibly to environmental change. They're thought to be faster explorers and less neophobic or fearful of novel objects. And they are thought to mount a stronger physiological or glucocorticoid stress response. And so those things are the physiological stress responses that they have are thought to be correlated to their behavioral responses as well. And so if I were to plot a theoretical reaction norm for a rural bird versus an urban bird, it might look a little something like this, where the slope for the urban individual is a little bit stronger. When they face an environmental change, they might respond more strongly. A lot of this work has been done at the adult level, and so it's really unclear when these differences arise and how they arise. And so in my lab, we're trying to tackle that. European starlings have altricial young, like a lot of other birds, and so their coping strategies for environmental stressors change really, really rapidly over just a few weeks when they're developing in the nest. And so at first, the priority is to grow, grow, grow. And anything that could detract from your growth is bad. And so individuals are thought to be non-responsive to stress, and they have a dampened stress response physiologically because it could impair growth. But within about a 2-week period, individuals are nearing independence. They're almost ready to leave the nest. And having some kind of coping strategy when you encounter a stressor becomes critical, because outside of the nest, you are going to encounter stressors that you have to respond to appropriately. And so here I'm predicting that the coping strategies or the coping styles that we see in adult birds might be shaped as early as in the nest box over the course of developmental age. And so we might expect that a nestling might not respond very strongly to stressors when they're really young in the nest box, but as they reach development or independence, nestlings from urban sites might respond more strongly. And so I sampled nestlings at two time points during the peak of their growth and when they were nearly independent right before they leave the nest box. And I looked at a few different behavioral coping strategies. One is their breathing rate, which is here on the y-axis. And I've got nestling age on the x-axis, where the first time point is at the peak of growth, and the second measurement is when they're almost ready to leave the nest box. And I've got the rural and urban nestlings separated by color. And you can see that each line connects the data points for one individual nestling. And so on average, what I found is that breathing rate was higher for urban nestlings, which is correlated to their behavioral stress response. But this didn't increase with age like I expected. A second measure that I looked at was the number of handling struggles that nestlings perform when they're held on their back. And so here, what I found is that at the peak of growth, neither rural or urban nestlings struggle very much. But over developmental age, when nestlings are reaching independence at the urban farm, they do increase the number of struggles. And so this supports my hypothesis that individuals at the urban farm do react more to stressors as they near independence relative to the rural farm, where nearly independent nestlings do not show this response. And so going forward, we're really interested in mapping on behavioral strategies or coping strategies in the nest box and how they match on to behavioral strategies once individuals have left the nest box, to see if there's continued development of coping style or whether these coping styles are representative in the nest box of the adult phenotype. We're also really interested in understanding how different early life conditions shape coping style both in and out of the nest box. And so, for example, the parental behavior that individuals receive or the quality of diet that they receive in these urban and rural environments might shape their coping strategies. Other aspects of their early life environment, like just being brought up in an urban nest box versus a rural nest box, might influence this. This could be related to noise, for example. And then genetic factors probably influence this as well. So we're gearing up to do some cross-fostering experiments across these environments to disentangle all of these early life effects to understand what creates a more flexible individual. So with that, I want to acknowledge everyone who's helped collect data on these projects that I shared, especially my collaborators, my lab field assistants, and everyone who's given me permission to work on their land. I want to thank the sources of funding that have contributed to this work, and also all the organizations that permit us to conduct this. And hopefully, if we have questions, I'd be happy to take questions, or if we have time for questions.
Thank you so much.
I think for questions, what we'll try is to have everyone just let us know that they have a question in the chat box, and then we'll in order that way.
Oh, Eric seems to have a question.
Do you want me to look at the chat? Sorry, I haven't found it.
Eric.
Well, I was just wanting to say that that was a fantastic presentation. I'm sorry that my reaction became framework for a question. I don't have a stunning question yet. I'll work on it.
Thank you.
I lied. Can I ask a question?
Sure.
I guess I want to frame it as a comment. I really, first, I came in a little bit late because I was teaching a class, but I really like the work on the group size.
Mm-hmm.
And I think that the literature of actually figuring out some of these like classical optimal group size problems and the like Sibley conundrum that we'd forgotten about for so long, it was really nice to see the costs and benefits. And I'm fascinated by the fact that there's this tendency to have too big groups, but there's benefits to being too big, as though too big is a good thing. I guess there's not really a question there. If there was a question there, do you have any idea why folks hadn't thought to do the nice job of quantifying it like you had for so long?
That's a good question. I think that the literature on sort of social benefits is not super clear or easy to sort of pull together, just because I think different social systems are social for different reasons. And so like it takes a lot of understanding background life history and natural history and ecology of a species, like our long-term study in superb starlings, before you can actually start to disentangle those things. And it will differ from one system to the next. So I know what you're saying. I think it's like confusing sometimes to identify the specific benefits of group living. And it does take like a long-term data set where you've got a lot of natural history information about a specific system. So I think it's just hard to do.
Are there any other questions?
Oh, there's something in the chat. Jack.
Hi, Sarah. That was a unsurprisingly great talk. So yeah, I have lots of questions about your work because we know each other, but I'll ignore the squirrel stuff for now. With the superb starlings, when there are like these different benefits to different group sizes, how does group size change for a given population? Like, is it just survival and mortality, or do they like move between groups? Like if I'm a starling in a group that is too big for this current situation, can I change that?
That's a good question. So the group sizes change with two things. So individuals can be born into a group and recruit into the group that they were born in, and they can die. But superb starlings do have this one dispersal event that they can also choose to do. And so when you're sort of a young, maybe 2 to 3 year old starling, you can choose whether you're going to stay in your natal group or you're going to immigrate to a different group. But after that one dispersal event, once you've recruited into a group, if you do choose to leave your natal group, birds tend to stay in their social group the rest of their lives. So it seems to be this weird thing that is a one-time decision, and once you've decided, you've committed. And then we also think, we don't know a lot about this, but we also think that there's a certain negotiation that happens when an individual wants to immigrate into a group. We do think that the group does have some control over whether they accept an immigrant or do not accept an immigrant. And some reasons why you would accept immigrants is if your group isn't big enough yet, or the identity of that individual. So sometimes individuals will immigrate into a group where like one of their siblings that's older has already immigrated into that group. And so it creates this sort of kin substructure, even though there's a lot of immigration in the system. Those are the two main ways. You can immigrate into a group or you can recruit into your natal territory. So group sizes do shift through time. And over the course of our study, some of the smaller groups have been like 15 individuals, and they might have grown up to nearly 30 and like fluctuated back down. We don't have a lot of clear information on what drives changes in group size over time. It doesn't seem to be that tightly correlated to rainfall as far as I know. So group sizes change. We don't understand a ton about the dynamics. And I think part of that is just because there's so few recruits available in any given year. Nest predation is so high, so there's so few individuals that do make it to independence where they can choose to stay in their natal group or immigrate. And so we just don't have that many observations of those decisions happening. Dave has a question.
Hi, Sarah. Really nice talk. I really enjoyed that. I have a question about the relationship between group size and longevity in the superb starlings. Just trying to think of different ways to explain that. Is there a difference in how long breeders live versus non-breeders? And related to that, does the proportion of breeders in a group change as the group size gets bigger? I would sort of expect, if I had to make predictions about those questions, I would expect that breeding is costly and therefore breeders would not live as long as non-breeders. And similarly, that as group size increased, there would be a lower proportion of individuals acting as breeders. And if you put those two things together, it may explain or help to explain why you would see this sort of relationship between longevity and group size. Do you have any thoughts about any of those points?
Yes, I have a ton of thoughts because this is exactly what I started wanting to study during my PhD. And then there are a couple things that really complicate looking at those factors, like cost of reproduction. One is that the starling breeding rules are fixed for one breeding season, but they change through time and over their lives really dynamically, like more than any other cooperative system I've seen in birds. So it's not a situation where once you acquire a breeding position, you are the breeder and you breed every year until you die. Our breeders and our helpers both take a lot of breaks where they sort of are part of that 50% of the group that doesn't do anything in a given season. And so it's really hard to observe cost of reproduction because I do think that individuals take breaks when they need it. It's also hard to look at the lifespan difference of an individual based on their breeding rule because the breeding rules change so much, and I've not been able to find a correlation between lifespan and like the number of breeding seasons you might have actively bred. I think individuals are just really good at taking breaks when they require them and then getting back into the breeding pool when they're ready to do that. So cost of reproduction have been really difficult to study. There's a second component to your question. Oh, the proportion of roles per group tends to scale up. So like the proportion tends to be the same across group sizes. So let's say 10% of birds in the group tend to be breeders. That tends to be true regardless of group size. So there are more breeding positions available in a bigger group. And there are potentially more there are more individuals fighting for them, but it's the same percentage. And so we've not found that fluctuates with anything other than rainfall. So when there's more rainfall, there tends to be more breeding, but that's the main thing that shifts percentages of roles.
Okay, thanks for that. That was really helpful. And it created one little follow-up question for me, which is what I was just, Oh, I've lost my train of thought here for a minute, but I'll get back to you.
Yeah, no problem. I'm also happy to answer questions via email. If things pop up in your brain tomorrow, that works too.
It just popped back in. So thinking about the longevity versus breeding status, I can understand why that would be difficult because they change breeding status over time. But is an individual more likely to be killed, in particular through predation, during times when they are acting as a breeder? Because I would expect that they might be more vulnerable when they're sitting on a nest, for example, and they might be more likely to engage in really close encounter mobbing behaviors, all of which might make it more likely that they'd be killed as a breeder versus as a non-breeder. Any thoughts about that?
Yes. So one thing that sort of prevents that from happening is during incubation, especially, other individuals from the social group guard the nest, even when like the mom is on the eggs, for example. And so I'm not 100% sure that incubation is more risky for the mom than it is for anyone else who's perched near the nest. And the other thing about differences between breeders and helpers is that they don't invest, like each individual doesn't invest that much in offspring care per se. And so it's not like the mom is working like 10 times as hard as the other individuals as far as finding food, or she's not that much more likely to mob than other group members. So during a mobbing event, it's really hard to see who's where. So I don't know if the mom is more likely to mob closer to the predator than other individuals in the group. But as far as other parental care behaviors that we do observe, the mom does a little bit more, but like not on any level more drastically than other individuals. So all individuals seem to care similarly. The mom might work a little bit harder, but it's not a very drastic shift. And then if we do try to look at like season to season survivorship, where if you bred in one year, does that influence your likelihood of coming back or breeding the next year? We don't see any patterns. And so I think that costs of reproduction probably do happen, but they probably happen over the whole life of a bird, not just from one season to the next. And so it's just incredibly difficult to measure. But I have a collaborator in math that I'm working with at Kennesaw, and we're trying to model some of this stuff because it's just too hard to actually measure empirically.
Okay, thank you very much.
Mm-hmm. Kelly has a question.
Yeah, so thanks. That was an amazing talk. Super cool. What I found really interesting was that in the squirrels, the mothers are kind of looking at the conditions and then inferring density and then passing, that information on. Have you looked at it, or do you see any kind of variation in the ability of the mothers to detect the density? Like, are they ever wrong?
Yes, that's a good question. And that's one sort of proximate explanation for why some moms might respond inappropriately. They might just be estimating density inappropriately or something like that. I don't think very good data existing in the long-term data set that I could use to tease that apart. There's also a little bit of variation in exact density within like the buffer of a particular squirrel's territory. So I've used like their acoustic environment because they use rattle calls to defend their territories. But it's possible that like some squirrels have a smaller buffer than others, and I've not tried to figure out how you would even try to determine what an individual's optimal buffer of how they determine the density around them specifically. So I don't have a ton of insight for that, but that is one possibility. And there would be really cool playback follow-up experiments that you could do to see whether some individuals can just discriminate density better than others. There might also be additional cues besides vocalizations that some squirrels rely on, which might be less reliable, and some cues might be more reliable. So individuals might differ in what cues they use. I don't have a whole lot of insight to differentiate between those possibilities, but they're all very likely reasons why a squirrel would respond inappropriately, especially if it's a costly misresponse to your environment, if that makes sense. So it's probably a mistake that they're making, if anything.
Thanks.
Yeah, another question. I think this is Levi, the MUN BGSA.
Yeah, thank you again for the talk, Sarah. That was super interesting. I'm asking a question about the starlings, and I'm sorry if I missed something, but I'm just wondering. So the breeders and the helpers both have this response in their glucocorticoids to group size, where it's lowest at an intermediate group size.
Mm-hmm.
But I'm just curious. I mean, I don't know much about like the difference between breeders and helpers, but like as I understand the last question you just answered, their care is similar for offspring.
Yeah, so it varies from one individual to the next. If you look at the average for breeders and the average for helpers, they're not that far from one another. So the breeders do a little bit more work, but it's not like the breeders work 3 times as hard as the helpers or anything like that.
So I understand there's some thought that maybe baseline glucocorticoids change with like your parental care or your breeding status or whatever it happens to be in different species. So is there I noticed there was like a shift where like the breeders and helpers had kind of different high levels of glucocorticoids at their lowest group size.
Mm-hmm.
Is that something that is I mean, is that something that like I guess is that something that's just a kind of artifact of the data, or is there a pattern there that's
Yeah, that's a great question. So I think I understood everything. Your audio is a little bit choppy, but I'll repeat the question and then you can give me the go ahead if it's correct, just so everyone else can also hear. I think the question has to do with the baseline glucocorticoid relationship with group size in the starlings and why the optimal, the sort of lowest baseline level for the breeders was a little bit lower group size and for the helpers was a little bit larger group size, and how offspring care might play into shaping differences in glucocorticoids. Is that the question?
Yes, correct.
Okay. Yes, I have thoughts about that. So I think that for I've looked at relationships between parental care and glucocorticoids in the starlings, and I've not found anything with individuals caring for offspring more having different glucocorticoids as a response or anything like that. The one pattern that I have found is that individuals that have higher baseline glucocorticoids prior to breeding, during the sort of early incubation phase, do guard more once the nest hatches. And so in that way, the only thing I found is that glucocorticoids seem to prepare individuals to invest more in guarding. That's the only relationship I found with any sort of parental care or behavior during breeding in the starlings. I think that one explanation for why the optimal group size as far as having lower baseline glucocorticoids was lower in breeders, I think it was like 30 or 35 birds, and in helpers it was closer to 40 birds. I think that one explanation for that is the way that breeders and helpers experience competition within their social group differently. So breeders probably get a lot more trouble from other individuals in their group who also want to take over their breeding role. And so I think breeders face more competition within their group during the breeding season, and helpers, because they don't have a commodity that other individuals want, probably face reduced competition. And so I think that's why the optimal condition of a helper might be a slightly larger group than for a breeder, because they just don't face the same kind of competition from within their group. Any other questions? Jack.
Hi, I can't stop, won't stop. With the predator and the nest guarding in the superb starlings, Is there other environmental fluctuation that would affect predator abundance or density independently from rainfall?
Yes, probably. So we do know that like the small mammal communities at the research center where I work also fluctuate with rainfall. And we also know that like a lot of birds of prey also fluctuate with the small mammal communities, which fluctuate with rainfall, probably with some kind of time lag. But rainfall would probably be one of the things that would influence one of the predators. The problem with the starling system, especially with the nest, is that everything would eat them. And so we get like small rodents, we get monkeys, we get mongoose, we get all kinds of snakes, we get birds of prey. I'm probably missing a few. We get squirrels and stuff. I think I said rodents. Anywho, there's so many different predators that would eat a starling egg or nestling that it's really hard. And I just think that there's not one clear environmental factor that would influence all of them the same way, if that makes sense. So I've had a really hard time trying to like retroactively figure out ways to estimate predator communities that might have changed over time in the course of our study, because we just can't monitor all of them. For the adults, it might map onto rainfall a little bit better because the birds of prey would definitely cycle with rainfall loosely to some extent, but I'm not sure, and I'm not sure about the snake communities, and I'm not sure that anyone's done all that thorough of snake surveys at the research center, so I don't really have data for that.
Yeah, I guess like if 90% of the nests get predated, then the odds of like one year being particularly bad for all of those predator species is like not very likely. Do adults die from non-predation stuff?
Yes, I think that they do. I'm trying to think. Maybe someone has brought me one carcass of a starling that was kind of on the ground with no clear wounds or anything, but it's all very anecdotal. So like I've also gotten like a head from a bird of prey that dropped just the head. So people bring me back starling pieces. We've seen that one starling getting eaten by a snake, but we just don't have nearly enough information to try to figure out like proportionally cause of death of starlings. I just know that predation is possible and likely. Disease and old age are also likely to happen at some point, but we have no sense of how to estimate relative importance of each of those.
Last chance for questions. Great. Well, yeah, thank you so much, Sarah. That was really interesting. So before you all leave yet, we were hoping to do like kind of a discussion. So if you want to stay behind and you want to talk to Sarah a little bit, you can turn your camera on, you can stay behind, and you can ask Sarah and she can ask us any questions, be it about her research, about her university or her career path. And yeah, so thanks.
Thanks for having me.
We have to get someone to show us how to do the clapping thing. Because we were trying to figure that out because it's a nice way to, oh, there you go. It's a reaction. Oh, okay. Yeah, I thought raising your hand would be also a reaction, but I think it's different. But also people could just give like a thumbs up maybe, or clap when they want to ask a question. Someone will know.
Yeah,
So,
I thought there was a raise hand function on Zoom, but I'm mistaken. I think it's the other platform I teach on definitely has a raise hand function, which I use all the time.
I think, God damn it, camera. There is one, but it's like not in the reactions, it's in like the participants.
Oh yeah, you're right.
You can go into, but it's like kind of hidden, and I don't think anyone really uses it.
We tried to find it, like before we started our seminars, we did a practice and we tried to find it. We looked it up, but I don't think any of us figured it out. I don't know.
I saw Dave do it.
Oh, Levi, you're cutting out. We didn't hear that.
Sorry, my headphones are super weird sometimes. I saw Dave do it when he had a question, which is, but I can't find it as I look through the participants.
We'll figure it out for next time.
Yeah. Anyway.
So,
yeah.
I have a question.
Yeah.
I have two questions. They're kind of peripheral to your main story, so I didn't want to dilute the other questions with them. First, it was really enjoyable listening to you, partly because you have such rich natural history component and experiences to both of the squirrel and the superb starling stories. I have one question about body size and body size variation. Both of these are reasonably long-lived species, in which, in these populations, the adults, in these populations, then the surviving animals actually, as individuals, persist through variable years that vary from one another, but also they're born in years of differing challenges environmentally, right? So the squirrels grow quickly in some circumstances when they're pups, and less so when they're not. So there's all this, and also with the starlings, I guess, correspondingly, there must be inter-year variation in fledgling size or weaning size. Does this translate into higher variation using a simple metric, just like a coefficient of variation of body mass or toe length or whatever, in those populations? That's my question.
Relative to animals that don't experience so much environmental variation from year to year for their early life, you mean?
Yes, and animals of comparable longevities, yeah.
Yeah, so the squirrel literature, I feel, probably has the data set to do that. I'm not sure if the team has looked at that before. Jack can correct me if I'm wrong. Variation in body size.
Well, body mass would be a poor metric, but a linear measure would be useful, or skull size,
Yeah, like hind foot length or something like that. I'm not sure if like early life environment has been mapped on to variation in size of different cohorts, or like coefficient of variation in cohort.
Well, from good years, for example, with the squirrels, when pups grow more quickly and presumably wean at a higher, greater body size, are they larger as adults than one-year-old animals who were born in poor years, for example?
That I don't know off the top of my head.
Hmm.
Do you know, Jack?
I don't think an effect persists into adulthood. Faster growing pups in the nest are bigger like at weaning and into the fall, but I think by the time the next spring comes around when they're a year old, there's no longer a difference. And also the growth rate and like the quality of the year is kind of weird, because in like mast years when there's a ton of food, they grow the slowest. And they grow really quickly in post-mast years because it's a competitive environment. But like there's not actually that much available to them in the environment once they get out there. So it's a little confusing.
Mm-hmm.
Well, there's certainly some species in which variation in body size, adult body size, is amazingly high for unknown reasons. I mean, a rather obscure example that you probably aren't familiar with is the least seed snipe, a South American species, which is crazy. They're like different species. If you look at a museum drawer at the skeletons, and I always thought, well, maybe it is a situation like what you faced that generates such high adult variation, unrelated to geographic variation. But anyway, those are just loose shotgun kind of questions. Another question I had struck me was one of your later slides of the superb starling, there's a little flock on the ground, and it looks like one of the birds is protruding its eye, and the eyes are obviously really important in optical communication. They're stunning, right? So it's got it back to the viewer, and its left eye looks like it's sticking out. Do they protrude their eyeball?
I don't know. I don't think that they do. It might have been the angle, like if the bill or if it had something in its mouth that was
Right,
like
right.
a camera that was sticking out. What I do know about their eyes is the color of the iris changes over their age, which is really cool. So it's actually one of the ways that we age them when we do get an immigrant that we didn't band in the nest. So sometimes they come from a social group that we don't study and band all the nestlings for.
Right, right.
Start really dark brown, and they fade to that like white, really bright color by the time they're about 2 years old. But that's the only thing about the eyes. I don't know that they protrude.
Well, the same is true of white-eyed vireos with respect changes with age. It's too bad Pierre Paul isn't here because he would know all that stuff. But anyway, well, those are my questions. Thank you. Everybody really enjoyed your presentation. And as the old guy on the block here, I must compliment you on your open and highly relaxed manner of presentation.
Thanks.
It's not, it's, yeah, it's rare, so good for you, apart from just the content. Okay, thanks. Well, I'm going to sign off. I have to meet a student. So have a good trip back.
Yeah, I'll be very safe in the airport. Thank you so much. Thank you.
Okay, bye-bye.
So Sarah and Jack, have you worked together at Windsor?
Guelph, we were both, I was doing my master's on the squirrels when Sarah was doing her postdoc. So we are old friends.
So it is a small world.
And Jack is much more of a squirrel expert than I. Sometimes people ask me a lot of squirrel questions that I'm like, there are hundreds of squirrel papers. The team is so prolific. It's really hard to keep track of all of the results that they found from this one system. So sometimes I get overwhelmed.
Yeah, it's a lot. I don't know why it like sticks in my head as like trivia points, but for some reason I can remember squirrel facts that I absolutely cannot about caribou.
Yeah.
It's too weird. The like undergrad you have right now working with starlings, that was like a cool project. Do you have other students doing stuff? Like what's current things looking like for you?
Yeah, I can tell you about everyone in the lab right now. So I just have my first 2 grad students who are starting, they started this August, so they're still developing projects. But one of them I think is really interested in how, so in European starlings, there's a similar maternal effect where moms can pass down glucocorticoids through the yolk of their egg into their offspring, and then that can influence their growth in the nest box and then map onto their phenotype for responding to stressors in adulthood. And so she's really interested in sort of mapping glucocorticoids from the mom into the independent fledgling, like through the egg, and connecting all those points basically, which hasn't been done super consistently. Like people know a lot about how moms pass things into the yolk, and they know about how yolk influences nestlings and how nestling influences the eventual adult coping strategy. But she's interested in mapping all of that continuously. She's also interested in how glucocorticoids might contribute to variation in sex ratios in different environment types. And so sex allocation in a favorable versus an unfavorable environment is kind of a rich history in the literature. And glucocorticoids are one mechanism that might map what is happening in the environment to the sex ratio of the brood. So that's probably where she's thinking about going. Another student I have is really interested in variation in diets, especially in urban environments. And so a lot of really bold individuals in urban habitats are more likely to eat foods that are maybe not advantageous to feed on. And so she wants to do like presenting different food items to individuals in rural and urban sites to compare like how willing individuals are to feed on non-natural food sources and whether they use those to feed the nestlings. one result I cut out is that we found that the urban nestlings increase in cholesterol a lot over development, but not the rural nestlings. And so this is sort of the jumping off point to explore like what differences in behavior along an urban to rural gradient causes that pattern in the nestlings. And I really suspect the parents are making weird decisions, and then understanding the consequences of those decisions downstream for the health of those nestlings and their growth and so on. So I think she's heading that way. And then I also have 4 undergrad students currently working in the lab. Jasmine, the one who I shared her results, is like a stats pro for an undergrad student. She approached me having taught herself R, and she's like, I really want to learn random regression models. And I was like, awesome. So we sort of developed this project on animal behavior, and she's doing really well. So she's actually going to start trying to write up a paper on guarding behavior, which I'm super impressed with her for. I have another student who is doing, she has an anthropology background, and one of my collaborators works with bonobo apes at a captive center, and they do like language acquisition in non-human primates, which is really cool. But they had this sort of situation where a zoo was going to donate or relocate an animal to their center, where the social group was going to change entirely, but not in a natural way where individuals accept an individual into their group, but where a researcher plops a new individual into your social group, and then you have to deal with the consequences of that. And so we've been collaborating with them. They've collected all these urine samples sort of during those introductions and then post. And so she's analyzing cortisol to look at how that changes with this like artificial sort of captive change in group dynamics. And they've also collected a ton of behavioral data on like affiliative and competition type behaviors between the individuals over that period. So we're going to have this long-term cortisol data set that she's running lab work with me on right now. And then I have a student who's using Nest Watch data, which is from the Cornell Lab of Ornithology, where people like community scientists can upload data of whatever's going on in their nest box. And so we've got this sort of North America-wide data set for European starling nest boxes. And she's interested in looking at whether the timing of breeding is related to environmental variables or the degree of urbanization of a habitat. And so the hypothesis being that most birds, when spring starts earlier, breed earlier. But like in the South, like in Georgia, I mean, it's nice enough that they could breed most of the year, but they don't. They stick to the season that like northern latitudes also breed at. And so I'm really interested in like if there are any factors that predict how flexible laying date is at some latitudes and not others. So she's exploring that. And then the 4th student is going through a lot of behavioral videos that I collected during my COVID field season, where I filmed the adults at nest boxes. And so she's looking at all these provisioning behaviors across different sites and how males and females share offspring care across different sites. So she's going through processing those videos. So that's what we have going on for now.
That's awesome.
A lot.
Yeah, it feels kind of nice now because we also have lab meetings. Your first year is kind of weird because you don't really have that much up and going yet at the university where you're at. And so it's kind of nice now. The sort of dynamic of lab meeting is everyone's got their own thing going on. They know more about it than me finally. And like, it's really nice to get updates from everyone in that context.
Do you have any data so far on the egg yolk glucocorticoids stuff? Nothing like that.
No, I haven't collected any of it, but other people have that I know in European starling. So there's a fair amount of literature on it already that we can use. I've never tried it, but you drill a tiny hole into the egg. I mean, the eggs are small. It's not like a chicken egg. You drill a tiny hole and then very precisely with a needle, you go in and you can sample the yolk. You can inject hormones into the yolk so you can do manipulations too. It goes both ways. And so every time I work on that protocol with my students and we sort of are planning what we would do, they're like, are you sure that we can do this? I'm like, nope, but other people have done it, so it can't be that bad.
Might be possible.
Mm-hmm. Yeah. Can everyone give me a little blurb about what they are doing right now? That would be super fun for me.
Sure. Who's starting?
I can call on.
Sure.
You can.
What's that?
Do you want to start?
Oh, sure. Sorry, my volume is quite low. So, I'm working on elk in Manitoba, and I guess my project started out doing like mostly movement ecology and just looking at differences in behavior and stuff in so the elk that I work on are in this kind of an agricultural environment. So they've got some natural habitat in like a swampy area and then like lots of farms. And so I was looking at differences in behavior, but part of that was to look at like stress levels and reproductive success. So I've been collecting data on that as well. And I've gotten way more interested in the glucocorticoids. So I'm collecting fecal pellets and looking at that. So I'm hoping to like get at like individual glucocorticoid levels and get some like fine scale, I guess pairing that with their movement and habitat use on like a very short term time scale and see if individuals differ in their glucocorticoid levels.
Cool.
So yeah.
Super interesting. Have you started looking at that yet, or are you still collecting samples?
I just collected actually I got to do fieldwork this summer, fortunately. So I got a second round of samples. And I have the data back from the first ones, and it looks like I don't think I have enough yet to tell anything. Hopefully more after this year, but I guess the one thing I did notice is like the seasonal variation in glucocorticoid levels. Like it's like I've been collecting in the winter and then during their calving season too, and they're like 4 and 5 times higher in calving season than they are in the winter. It's crazy.
Oh, cool. Yeah.
Yeah. But nothing on the movement yet.
Oh, no, that's interesting though. Neat. Do you want to go, Kelly?
Sure. I'm doing a master's in, I'm looking at brown trout invasion in Newfoundland. So brown trout were introduced in the 1880s and like near St. John's, and they've kind of spread westward. And so I'm looking at and kind of modeling like how far they've gotten, and then what like habitat characteristics allowed them to invade. And then I'm also looking at how they're affecting native salmonids or salmonids. So I'm going to be looking at the diet of brown trout, Atlantic salmon, and brook trout. And I'm going to see if like they're overlapping and see if there's like a potential for competition. So I'm in the field right now. We're doing a lot of electrofishing and stuff. So it was delayed, but we were lucky enough to get out, and it's going well.
Cool. Neat. Do you like electrofishing? So you're trying to get any of those 3 species you mentioned?
Yeah, exactly. So we do like river surveys in controlled areas, and then we take a subsection for diet. I like electrofishing. It's kind of backbreaking work. Like it's very, and it's very limited. Like you can't do it when it's raining. You can't do it when the water's too hot. And so we're really dependent on weather. It can be really frustrating. Like, you know, you're out there and you have all the equipment ready. You're allowed to do it. Like COVID's not a factor anymore. And then it's too hot or it's raining. Like you can't do it. So it's kind of frustrating at times, but I do like it.
Cool. That's what I've heard. I've heard a lot of people standing around and not being allowed to electrofish, and that's electrofishing.
That's basically it. Yeah. But it's cool because like I have other stuff I'm trying to do right now. Like I'm trying to write a paper. So on days we can't, like today we're not electrofishing. So I'm kind of like writing my protocols, trying to get stuff going. So I'm okay.
Oh. Neat. I have Jordan next. I don't know if you want to introduce yourself. Hi.
Hi. Great talk. I joined a little late because of other things, and I've been in and out of the listening. So I'm doing PhD. I'm in very different than everybody else who's right here. So I'm in microbiology.
Mm-hmm.
So I look at antimicrobial resistance mainly in the environment and wildlife. So I really couldn't do a whole lot while I was home, just analyze some data, wrote a paper. I've been back for almost 2 months now, which is nice. So just doing molecular, just doing running PCRs every day. And I'm starting up like projects looking at avian influenza and how they do they express different types of cell death for different strains. So we're just starting that up. So I had to go check our cells partway through, make sure that they're doing okay.
Cool.
Yeah. So.
Cool. I spent a lot of time trying to convince people that avian influenza is not too big of a problem in the birds that I studied.
Yeah.
Do you have any preliminary results so far?
From that stuff, so I was involved with a project with the US government where we sampled ducks for influenza. And then we split samples, and half was screened for influenza, and the other half was put in filtered water, and then put in a barrel at the bottom of the lake for all winter. And then in the spring, we retrieved the samples and screened for influenza again to see if we can detect viable virus after they spent all winter. And then if they detected the virus, they tried to culture it in the lab. And those that they cultured, they actually infected baby ducks to see if it was actually infectious. Yeah, so that paper that I was part of just got published on the 9th, so Wednesday. And it looks like the environment can serve as a reservoir for influenza reinfection the next year. So birds that excrete it during that breeding season, they can get in the environment and then serve as a reservoir and infect birds the next breeding season when they come back to the wetlands. So it's kind of small scale, but it's a concerning issue.
Yeah. Are there any sort of practical things that we could do to treat water?
And that's some of the things they spoke about in the paper, but it's hard because, well, let's change the total environment to try to get rid of this. But temperature has a factor, so the colder it is, generally the more viable viruses. And pH is a big deal as well. But like big ecosystem changes is probably not really.
Yeah. I mean, we can dump bleach everywhere, right?
Could do that, yeah. But
cool.
Yeah,
Thanks.
yeah.
Jack, do you want to give an update about what you're working on? I feel like I see you semi-regularly, but I actually don't really know that much about what you do.
Yeah, I was just thinking that. See if my webcam actually stays on for a while. It's a bloody laptop. So currently with the Fogo Caribou project, I'm kind of in literature review mode as my field season plans for the summer were scrapped. So hopefully I'm going to get out there in November to do a bit of behavioral observations and filming. But kind of the first chapter I'm looking on is sort of calving site fidelity and habitat selection. So like where are they choosing to give birth? Are they like returning to the same locations year after year? Because that seems to be a thing in a lot of ungulate species, but not all of them. And also a lot of it is attributed to predators, which like there aren't really that many of on Fogo. It's kind of a weird place. And whether or not there's like a social site fidelity, which is this thing that I think is sort of made up, and I don't know if it will happen, but like, do they select for a social environment in addition to a physical environment of like their friends near them? Because they form like calving herds. So right now I'm going through an Excel list of 400 papers about site fidelity to see what I can find, which is not as exciting as chasing caribou.
Yeah, it's got to get done. Cool. And how many people go when you're headed in November? Is it just you and field techs or something, or is it a big team that's headed out there?
Yeah, probably just me and one or two other people. If Mike and Yaz are here by then, they might join me if I can convince them. It's mostly whoever I can finagle from the lab to join me for a week or two. For like the calving season in May that when we're going out for, there's usually like around 10 of us, I guess. It's like the big push to get all the babies. But other than that, it's like 2 or 3. It's no squirrel camp.
That's nice though. And I think for COVID, that's probably better, having a small group.
Yeah, they don't want us to put 10 people in a trailer together for unsurprising reasons.
Yeah, for a while, folks were allowed to do research in teams here, but everyone had to drive to the field site in their own vehicle. So then it just was kind of, I mean, it's safer that way, 100% not complaining about that, but it was a little bit of a headache if you wanted to meet like 4 other people somewhere, and then you had to drive 5 vehicles there, figure out parking. It's just all kinds of different aspects of research. Cool. Thanks for the intros, folks. And thanks so much for inviting me. It was really nice. You have a really great department. Lots of good questions. Actually, a lot more questions than I typically get.
This was sorry, go ahead, Kelly.
The turnout was also really good. When it's in person, our seminars maybe get like 15 people. And so we had like, I think 20, which was really, really impressive. And I think it just speaks to your talk kind of dabbling a little bit in everyone and kind of everyone being interested. So yeah, thanks so much.
Cool. Is it all, your seminar series is always student run?
Yes, but yeah, so this is our I guess our grad student association for biology, and yeah, so we organize, and usually usually we all meet in person and we have a snack and everything, but yeah, obviously not happening right now. So this is like, I think this is our first virtual one too, and yeah, it went pretty well.
Cool.
And we're super grateful to have you. Like I'm already getting messages from people about how great the talk was and how excited.
Awesome. Thanks. No, it's so nice. I mean, because it kind of sucks for everyone too. Like when conferences are canceled and like at key career stages, like I don't feel bad for myself right now, but I feel extra bad for students who are like at a transition in their careers, and like a conference can really make a difference for networking. And I'm always super grateful for every society that puts on a virtual meeting, or for departments that put on virtual seminar series, stuff like that. I think it's really good.
At this point, people are starved for information and for interaction.
Yeah.
I feel like the one silver lining also is that we like get to open up our pool. Like we don't really have the funding to fly people to Newfoundland that much, so a lot of the in-person ones are like Mun folks. So it is neat that we get to pull in people we wouldn't have to otherwise.
Yeah, that's a lot of fun. I know I'm doing, so our departmental seminar is a little bit different here at KSU, but I'm running it this fall. And it's a senior course that like about 80 students registered for, but it also doubles as our departmental seminar where faculty and grad students would come too. And so it's been so much more fun because it does like free me from having to find people in Georgia. And there are lots of people in Georgia. So I'm super lucky because like within 2 hours driving distance, we have, I don't know, like 5, 6 great universities where you can invite other people. But like if you're thinking on a 4-year time scale, you're going to start repeating invitations a lot. And so I'm really excited that we can invite a lot more folks from all over North America. It's nice.
Yes.
Also, it's a lot of work to organize a seminar series. So go student team. That's fantastic that you guys are pulling this together.
Yeah, it's been, well, it's been an interesting year so far. So hopefully it'll keep going well. And yeah. But anyway, yeah, thank you again, Sarah. This was fantastic.
Yeah, awesome. I don't know if I have to go in about 30 minutes for some more meetings, some more virtual meetings, which is always fun way to spend your whole day. But this was definitely by far my favorite virtual meeting of the day. But yeah, I'm happy to keep in touch if y'all have questions, or if you want to talk more glucocorticoids, Levi. I know I didn't put that many into my presentation, and I felt bad, and I felt like I should have added more, but I'm always happy to chat with folks' research. So feel free to email me again.
Great. I probably will take you up on that.
No problem.
I have to go as well, but yeah, thank you so much again. And our 3rd seminar coordinator wasn't able to make it today, but he's doing some hormone stuff, so he might email you as well, because he was really interested and he was really sad to miss the talk.
Sorry. Yeah, that's no problem. Inbox is always open, for better or for worse.
Cool. See ya.
All right. See you guys.
Bye.